Hi everyone, this is Mr. Sinti, and this is our conversation today about the cell membrane. The cell membrane, interestingly enough, is sometimes called the plasma membrane. You might have heard that before because people own plasma screen TVs, and plasma just simply means liquid. And so that's a great name to describe the cell membrane because it is a fluid. These molecules that dominate in the cell membrane, the structure of the cell membrane, are what we like to call phospholipids. And if I were to draw one like this, right next to it, phospholipid, it has that circular area or head, and this area is polar. And what's interesting, it has these nonpolar tails. And the arrangement of a phospholipid is that of a bilayer. And so we have these two heads which are in contact with water on both sides. Water would be out here in the cell membrane, and it would be on the inside as well. And so these are the heads are in contact with water and the tails are not. What's interesting about the cell membrane, if I extend it around like this and make a whole cell out of it, if you think of the cell as the smallest living thing, here's the cell obviously, right here, and this is on the outside of the cell. You could literally say that the cell membrane is the edge of life itself. It's kind of dramatic, but it's true. And it's described as selectively permeable. Permeable means that some things can penetrate. If something is permeating, meaning like if you got ink on your shirt and it goes through, it means that it permeates through, passes through, permeable. But notice how it's described as selectively permeable, meaning some things are going to be able to pass through and other things are not. Sometimes it's even described as semi-permeable, meaning like a semicircle, half the things are permeable, half are not. And so as we go on in our investigation of the cell membrane, let's take a look closely at the structure of a phospholipid. This may look familiar to you. And so do you notice here this region of a phospholipid is what we like to call the polar head. So this part is polar, meaning attracting to water. And this down here is the hydrophobic area, or the tails. This is nonpolar. And so what's interesting about the cell membrane is that it's partly polar, partly nonpolar. It's made up of a glycerol, and remember if there were 3 fatty acids, it would be a triglyceride. So it's a curious lipid in that it's polar and nonpolar. And so when the consequences of this are that in order for it to be part of the cell membrane, it needs to be in a bilayer. And so it sort of rolls off the tongue, phospholipid bilayer. And this is what we mean by bilayer, meaning 2 layers. This is 1 layer, this is the second layer. And so again, this is the part that interacts with water, this is the part that interacts with water down over here. And if we go to the next slide, here you can see if you look at the whole cell membrane. Now this is a simple model of the cell membrane because the truth is the cell membrane is composed of more molecules than just phospholipids, but I think this is a good way to begin. If you think of the cell three-dimensionally as being surrounded by phospholipids, phospholipids are kind of like oil. They're not exactly like oil because if it was oil, it would be a triglyceride. And so this is a phospholipid bilayer, and our main point of looking at the structure of the plasma membrane is I'm going to start asking you questions about whether or not something can pass through. Remember how we said that the cell membrane was selectively permeable? Well, if I said that, would you be able to throw a basketball? Would you be able to throw a basketball through a fence? And the answer to that would be like, no, it's too big. But if you were able to throw like water across a fence, you'd say yes, it obviously would be able to go through. And that's because you know the size of a basketball, and you know what water is capable of, and you know what a fence looks like. So our intention here is if we understand the structure of the cell membrane, you'll be able to predict, I think accurately, what can pass through the cell membrane easily or with more difficulty, and that's our intention here. So we're going to start off with a phospholipid bilayer. But I mentioned that the phospholipid bilayer is the most abundant molecule is the bilayer, right here, phospholipids. But the truth is that there's proteins also embedded. And so as you can see here, there's a phospholipid bilayer, and this purple thing is a protein. And so proteins are embedded in the phospholipid bilayer. And so it's been described, the membrane, as a fluid, meaning that it's kind of oil-like, it has viscosity. But it's also been described as a mosaic. And you might know this from art, that a mosaic is all sorts of different tiles, and you put them together and it makes a picture. So it's a combination of phospholipids and protein that make up the plasma membrane. Let's take a look at another picture of that. So this is our current understanding of the cell membrane. It does look a little busy, but let's take it apart piece by piece, and let's take a look at that. We could say, apologize for the autosave, but right here we have the phospholipid bilayer. That's number one. And then number two, see these purple structures right here? These purple structures are proteins. Let me add another protein here. Now a protein, if you recall, is a chain of amino acids held together, and they form these interesting three-dimensional structures. So some proteins are embedded, meaning that they're integral. They can actually pass through the phospholipid bilayer. Some proteins, as you can see, this one right there, is actually only on the inside of the cell membrane, and some can actually only be on the outside of the cell membrane. So the proteins are a big deal. What else do we have here? Do you recognize these little hexagon-shaped things? Let me add them. These are little monosaccharides. And sugars can connect together. I wouldn't say that that's a polysaccharide. I would say this is a few sugars. I'd say these are oligosaccharides. Notice that there's oligosaccharides on the outside of the cell membrane. This is on the outside of the cell. This is on the inside of the cell, if I were to draw it around like this. That's curious. No sugars on the inside, but sugar on the outside. What's the function of oligosaccharides on the outside of a cell membrane? We believe that the sugars on the outside of the cell membrane help cells recognize other cells. Sort of like when you look at someone, you can tell who they are based on their outside superficial physical appearance. So this is what other cells would be seeing if they saw this cell. They would be looking at the sugars. So it's kind of curious. The sugar coat, if you will, and the proteins on the outside will determine whether one cell will recognize another cell. And that's actually rather important when you talk about how white blood cells in our body, their main function is to recognize foreign invaders like viruses and bacteria. That's pretty important. Let me point out another constituent of the cell membrane. This is something we've talked about before as well. Do you notice this little area that I'm highlighting here in yellow? This little molecule is called cholesterol, and it's a 4-carbon ring. And notice where it's located. It's in the inner part of the cell membrane, which is completely nonpolar. Cholesterol, which often has a bad association with it. People sort of like, and rightly so. Cholesterol can be harmful and cause heart disease, but what's interesting is that it has a good function here. Cholesterol is produced by the body, and it's embedded in the cell membrane. And what it does is that it helps the phospholipids from being very tightly packed. And if they're tightly packed, the cell membrane will become more of a solid and less of a liquid. So it prevents the tight packing of the phospholipid tails. Kind of complicated, but by preventing the tight packing, that prevents van der Waals forces from occurring, and therefore it's more fluid. And so therefore the cell membrane is able to move and be more dynamic. I will come back to this point later, because one of the functionalities of the protein are to allow molecules to pass through, and some of them, the proteins, act as enzymes, and you need that fluidity for that process to occur. Also, these little threads in here, I wish they would have been, the person who drew this would have used the same color, but these threads inside are also proteins inside the cell. And you're like, well, how come they're not globular like these? Well, that's the thing about a protein. They're very diverse in their physical appearance. These proteins inside can push and pull on the cell membrane, so if the cell membrane needs to reach out or it needs to pinch in when the cell divides, None of that would be possible if the cell membrane was more solid, and so cholesterol helps out in that way. So what do we have here? We have a mosaic. Phospholipids, we have proteins, we got some sugars on the outside, and we have some cholesterol. So a fluid mosaic. Yeah, that's what we're talking about, a fluid mosaic. And so let's continue on and see what the next slide presents. So who came up with this model of the cell membrane? Well, the truth is, like any major discovery or anything important in biology, it wasn't just one individual. Usually there were several scientists leading to this level of understanding, each contributing a little piece of the puzzle, so to speak. But the big paper came in 1972. These two professors, I think from the University of California, San Diego, published this paper in Science, and they called it the fluid mosaic model, in which they describe, as we have been talking about, the plasma membrane being a fluid dominated by the phospholipid bilayer with proteins embedded, cholesterol, and sugar on the outside. So it's our current model. Current meaning it's always subject to change. When we're able to discover something new, we'll simply add it. It's kind of neat. And so one of the things that I haven't mentioned, but I could, about the cell membrane is, in addition to it being a fluid, meaning that these phospholipids are kind of like oil moving around back and forth, they can move laterally back and forth, but they rarely flip-flop. In other words, they rarely rotate like this, and I think that might be obvious in the fact that the heads don't like being on the inside, and the tails certainly don't like being on the outside. So that sort of shows you that the configuration is polar and nonpolar. This is what I mean about the cell membrane being too solid, or maybe even being too liquid. You don't really want extremes on both sides of it. Like, say that the cell membrane was, in fact, made up of triglycerides. Let's say that it was. Do you remember that triglycerides, one of the properties of fat was that it's solid because it has saturated fatty acids? And one of the things about an oil is that it was liquid, and that's because it was polyunsaturated with all these kinks. And so what's interesting is the cell membrane is neither too solid nor too liquid. It's sort of a nice compromise in between, because the cell membrane needs to be fairly strong, and it doesn't want to break. But it doesn't want to be too solid because, again, it needs to be dynamic and capable of bending in and out. And so it's kind of interesting. What's fascinating about this is that the cells can make adjustments given certain temperature. You can increase or decrease the level of saturation. For example, if you're a cold water fish and the temperatures are obviously very, very cold in the ocean, you might want to go with more unsaturated fatty acids in your phospholipids. Therefore, you'll be able to maintain fluidity. And so what's interesting about that is people have sort of discovered that notion serendipitously by the fact that when people consume a lot of fish from cold water, like fish oils contain a lot of unsaturated fatty acids, and therefore they're slightly healthier as a result of it in terms of their heart disease. I think that's kind of interesting. The fact that organisms can adjust this is cool. And so I mentioned before about cholesterol also helping to make something solid or something liquid. Cells can also adjust the amount of cholesterol, whether it's too hot or too cold. Because a lot of cholesterol will prevent the tight packing, which will increase the fluidity, and then it can also remove some in order to increase fluidity if it's very cold. So you can make adjustments based on the degree of saturation of the phospholipid hydrocarbon tail and how much cholesterol you embed in the membrane as well. So basically the membrane is a collage of all different kinds of proteins and all kinds of sugars, and there's even proteins on the outside of cells between cells. Like, for example, if I draw this over here as being one cell, come around here, say there was another cell over here, and it's like, okay, there's space in between. That space in between cells, there's also protein there too. In physiology, we like to talk about that a lot. Those proteins help strengthen the tissue, and they're often dominated by a protein called collagen. So I just want to point that out, that there are extracellular proteins as well that connect to the cell membrane. So what's the story with these proteins? Well, what's interesting about it is the cell membrane, if the cell membrane was just made up of a phospholipid bilayer, and I mentioned how that can be selectively permeable, what's interesting about that is that some molecules can pass easily through a bilayer and others cannot. And so to allow passage in and out of the cell, proteins, can you see how this picture sort of looks like a door? It looks as if one of the functions, and it is, one of the functions of a protein is to allow molecules to enter in or out of the cell. It provides like a corridor or a little tunnel for molecules to pass through. You might be thinking, all right, perhaps, but why is it that we even need this in the first place? Well, let me show you this. I'll see if I can animate it. Here's a picture of just the phospholipid bilayer. Pretend there was no protein at all, and you were wondering what kinds of molecules were able to pass in and out. Well, what's of consideration is polar on the outside, non-polar on the inside. So something that is lipid soluble, in other words an oil, something like this you might predict would have an easier time passing in because it sort of like dissolves like. Let's check it and see. Yes, in fact lipids go through the cell membrane very easily. Now here's something, oxygen. Now oxygen is held together by a double covalent bond, and so this is completely non-polar. So if it's non-polar, that means that there's going to be no affinity for water, which means it's sort of like the cell membrane. It's non-polar, and so it's very small and it's very non-polar. So you might think, I have a feeling oxygen goes into cells very easily. And let's check it. It does. Oxygen passes through the cell membrane very easily, so it's permeable. Now let's consider this big green guy. Now green, glucose. You might be thinking, all right, monosaccharide, I remember this. It has a lot of hydroxyl groups. That means that water, as you can see, clings all over it. And it looks like it's polar, and it might have a difficult time passing through the cell membrane, but you might be thinking, I think the cells need glucose, and I have a feeling it's going to pass through. But the truth is, it doesn't pass through. It doesn't pass through the phospholipid, pardon me for that. It doesn't pass through the phospholipid bilayer. And even something small, like an ion. Now, this uses sodium, but it could be potassium, it could be chlorine, it could be calcium. All these things are crucial, but because they're electrically charged, it means that water clings to them. So check it out. Like when I push play, ions and polar molecules don't come into the cell. And you might be thinking, whoa, this is really bizarre. This is like, I don't know how that could be. Well, watch this. If I were to add a protein like this, let me do it really quickly. If I were to add a protein, now one of the things about proteins like enzymes, do you remember, they're very, very specific. So if I had something out here, let me use a different color. If I had glucose out here, what's interesting about glucose is that, remember, it doesn't pass in through the phospholipid bilayer, but it can pass in through a protein. So a protein can actually facilitate the movement, facilitate meaning help, can facilitate the movement of polar molecules and ions across the cell membrane because the phospholipid bilayer is impermeable. So that's kind of important. And so let me move that out of the way and come back to this. So this is, I like this picture. This kind of shows the three-dimensional structure of the protein. So proteins can penetrate the hydrophobic core. So this is a little advanced, but I'm going to say it anyway, of course. These amino acids in here, these amino acids in here are all nonpolar, and these ones are polar because they're on the outside. But what's curious is the inside tunnel is made up of polar amino acids, which means that if I had that glucose again, like this, and glucose was trying to get into the cell, no, can't come in. But can it go through a protein? Yes. It goes right through the tunnel and it can enter. So proteins can facilitate the movement of polar molecules and ions, like sodium, for example. Let's do that. I have to make another protein over here because these doors, these protein doors, are specific. So then sodium can come in like this. And then let me make another protein door over here. Let's say potassium wanted to leave the cell. Say this is the cell here. If potassium was going to leave, it cannot go through there because that's a glucose door. Can't go through there because that's a sodium door. It would have to go through its own door. But proteins can make all this happen. That's kind of interesting. Now, let me ask you this. What about oxygen, like O2? What happens if there's oxygen here? Does oxygen need to pass through? The answer is no. It can just simply move through the phospholipid bilayer. Here's another important molecule, carbon dioxide. You're like, this is also small and it's also nonpolar. How do I know that? Because if I draw it like this, these are nonpolar covalent bonds. Small nonpolar. What do you think? Right through the cell membrane. So gases, small gases, very easy. And then you're like, okay, that's interesting. What about perhaps one of the most important molecules of all, water? And you're like, okay, what's this going to do? This is small, this is polar, but it's so small that it's capable of just moving right through the phospholipid bilayer, either this way or that way. So that's interesting. Water can just pass right through the phospholipid bilayer. There will be, in a later lesson, an example of that I give that it recently discovered, like maybe in the 1990s, recently discovered there are proteins that will actually move water across the cell membrane very rapidly. And those were discovered in the kidney. It's kind of interesting. But for the most part, water moves right through the phospholipid bilayer. Let's take a look over here, the next slide. So here again is the phospholipid bilayer. This is a protein. These are the cholesterols. These green guys out here are the sugars, which are important in terms of recognition. You're like, well, is this the only function of a protein, is to be a tunnel that facilitates the movement of molecules in and out? And the answer to that is no, there's some other interesting functions as well. And so if you make a list, here's 6 additional functions of membrane proteins. Some of these are interesting. I mentioned the first one as being transport, so moving molecules in and out. This is an example of active transport. I will come back to this in a future lesson. But transport, materials across. And you're like, what's an example of what that blue thing would be? You would say, could be an amino acid because that's small polar. Could be glucose, could be fructose. It could even be any ion. But it wouldn't be oxygen, water, or carbon dioxide because that just simply goes through the phospholipid bilayer. Another function of a protein embedded in the cell membrane is it can even be an enzyme. Enzymes are often in the cytoplasm and in different organelles, but they can be part of the cell membrane as well. So substrates in, products out, speeding chemical reactions. This one's a little advanced, but I'm going to mention it anyway. Sometimes a cell receives a message in the blood, like for example a hormone. And a hormone can either come in or just simply knock on the door. And if a hormone binds to a protein in the cell membrane, it'll change its shape and cause the cell to do something or change, make something or not make something. Sometimes cells, this is one cell over here, and this is another cell over here. Sometimes the cells use the proteins to help connect. Like for example, part of the digestive system, the cells are really close together, like a fence. Like you don't want any food to be able to get in between. So they're all linked together with these proteins, holding them in and out. And then this function I mentioned before. See these little green guys? Those are the sugars. So this might be, let me erase so it's not too busy. This might be, right here, a white blood cell. And it's looking around. And this right here is a bacteria. And it recognizes this as a bacteria because, ooh, an unusual sugar arrangement. And so then the white blood cell would then go as far as to either eat that or release an antibody and destroy it. So that's kind of a cool function. And then this is the last one. The proteins can act as attachments, either to outside proteins or inside. So it's kind of interesting. If you want the cell to, let me draw a cell. If you want the cell to sort of pinch in, if it's going to divide, the way you're going to get it to pinch in is that there's proteins embedded here. And then these proteins are going to interact and grab a hold of the membrane proteins and pull on them. And so you can get cells to pinch in or move outward based on the attachment. That's kind of interesting. And then finally, I mentioned this before, but this is a cool picture. These things that are now in orange color are sugars on the outside, and they help to recognize one cell from another. One of the great examples of recognition is when you donate blood. Most of you know that each of us might contain a different type of blood. Like I might have A blood, and you might have B blood, or O blood, or AB blood, or positive or negative blood. What does that all mean? Like if you actually took a look at the picture of a red blood cell, you wouldn't be able to see it, even though this is under incredible magnification. These are red blood cells, they carry oxygen, but the cell membrane of a red blood cell actually looks like this. And so let me take it around like that and say, all right, here's a red blood cell, even though I've drawn it blue. The outside of the membrane has these different arrangements of proteins and sugar, and if you have a certain arrangement of sugars and proteins on the outside of your red blood cell, you're considered to be A blood. And if you have a different kind, you're considered to be B. And so when you donate blood, here's a bag of blood, kind of gross. This is O blood. And so it's the sugars on the outside that will mark the difference between A blood and B blood and O blood. And then, like for example, I'll just make it cartoonish. If you had, let me make something that contrasts. If you had A sugars and proteins on your red blood cell, like this, sort of like pizza toppings. If you had pepperoni, you'd be considered to be A blood. And then likewise, if you had a different sugar arrangement, you would be like B blood, like this. And predictably, I don't know if seemingly predictably, if you had A proteins and B proteins, you would be something like AB blood, like in other words, pepperoni and sausage, something like that. And so sugars are rather important in terms of recognition, self from non-self. So this is all I wanted to say today about the beginning of our understanding of the structure and function of the plasma membrane. I hope you enjoyed that. Thanks for watching it.